So it is a real pleasure to introduce our speaker for this afternoon, Dr. Ann Stevens. She received her undergraduate degree at UC Davis in biochemistry. She then completed MD, PhD from Baylor University in Texas, followed by completing a residency in pediatrics at Children's Hospital in Cincinnati and a fellowship in pediatric rheumatology at the University of Washington. She currently serves as the Program Director of Rheumatology Education at Seattle Children's Hospital and is an Associate Professor of Medicine at the University of Washington. Her research interests focus around the role that maternal cells play in the pathogenesis of systemic lupus. She has numerous publications, including articles in the Proceedings of the National Academy of Science, the Journal of Rheumatology, and the Public Library of Sciences Genetics section. Today, she is going to talk about her new research interest, and that is the role of oral hygiene in chronic inflammatory diseases. Please join me in welcoming Dr. Stevens.
Can everyone hear me okay? I'm really pleased to be here. Thanks for inviting me. I have a soft spot in my heart for PLU, and my stepdaughter here. And my godson, Nathaniel, is a student here. Nate the Great in the back there. Yay! And my very first research technician is a PLU grad, and she was fantastic. He worked with me for 5 years and now is a resident in dermatology at Jefferson University in Philadelphia. So I know you guys are getting a great education, and I hope I can contribute a little bit to it. I'm going to talk about my work relating oral inflammation to systemic inflammation, specifically arthritis. And I'm also going to I'm going to talk along the way about the various people involved in this project in hopes that you guys will kind of see what sort of jobs people do and what sort of teams we need to put together in order to enact these sorts of research projects. So here's the problem. This is what I do on Thursday afternoons in my rheumatology clinic, is mostly seeing children with juvenile idiopathic arthritis. And we call it idiopathic not because the patient's an idiot, but because the doctor's an idiot, because we don't know what causes it. We know there's a genetic component to this disease, but we don't know the gene. We know that infections can trigger the disease, but we don't really know what infection. And sometimes injury can trigger the disease. So we might see a little kid who fell down and hurt their knee, and then their knee swelled up, and then 3 months later it's still swollen. Now it's idiopathic arthritis. So there are a lot of mysteries to this disease. There's no cure. We have treatments that are anti-inflammatory treatments, but they're pretty toxic, and the kids have to be on them for years and years and years. This is one of the major causes of disability in children. It occurs in about 1 in 1,000 children. So it's as common as diabetes and cystic fibrosis and these other diseases that you hear a lot about. So that's our problem. And here's the novel hypothesis that I'm going to take you through today. That is, this is what triggers it. Infection in the mouth leads to inflammation in the gums and in the bone in the mouth, in the jaw, which activates immune cells, your white blood cells, and activates them to make antibodies, which then travel to the joints to trigger inflammation in the joints, which we call arthritis. So that's the whole talk in a nutshell. Where did this come from? I was studying, as you just heard, cells from mothers and children, how they may trigger inflammation. Studying T lymphocyte activation and systemic lupus erythematosus, which is a disease that encompasses arthritis, but also a lot of other inflammation in a lot of other parts of the body. So I went to our big national rheumatology meeting in 2009, and the plenary session was given by this guy, Gerald Weissman. So he is one of the old original rheumatology physician scientists. He made some of the original rheumatoid arthritis discoveries. He's the chief of rheumatology at NYU, a Guggenheim fellow. This is a guy you respect. So when Gerald Weissman gets up there and says, I think there's a link between gingivitis and oral inflammation and arthritis, then I was listening. And it was very new and very novel and very interesting. And what he talked about was partly epidemiology. So he said that in Europe and Asia, no one had ever heard of rheumatoid arthritis. until way up here in the 20th century. In Europe in the 16th century. In Asia and Africa not until the 20th century. So the first place in the entire world where we have evidence, archaeologic evidence of rheumatoid arthritis, and we can tell this from erosions in the bone that I'll show you, is in Alabama, in the New World. So rheumatoid arthritis is a New World disease. We have lots of archaeology in the rest of the world. When you think about Egypt and the mummies, we can find a different kind of arthritis in the mummies. It's called a spondyloarthropathy. We know the gene for it. It's a completely different disease. But rheumatoid arthritis was only in the New World until the Old World met the New World. When Columbus came over, we started exchanging people, organisms, genes. What is it that we were exchanging is the question. And then he talked about sugar and how sugar didn't start coming into Europe until the 16th century. And first it was just for the royalty, and then when it got to be more easily accessible to the people, everyone started putting sugar in their tea. And that's when cavities and oral hygiene started going downhill, and that's when arthritis started to be reported first by noting in medical journals by this Landry Beauveau, who first described what we now know as rheumatoid arthritis, and then first named later on in the 19th century. So it's such a common disease. We think it must have always been around, but it hasn't. So this suggests that perhaps there is something infectious that could be related and could be triggering this disease. Then in 1982, Snyderman and McCarty first commented on the mechanisms of inflammation that are very common in this periodontitis, which is inflammation in the jaw bone around the teeth, and arthritis. Both of them can start with gingivitis. Periodontitis can start with gingivitis and that spreads to the bone. It's self-sustaining inflammation, meaning you don't need to have a constant, what we think is a constant infection, in a compartment next to a bone. So in the joints, it's the synovium that aligns the bone, and in the mouth, it's between your gums and your teeth. And you see inflammatory cells and other phlogistic factors, so there's like cytokines and antibodies, that lead to this pain, swelling, and tenderness, and then erosion of the bone. So it's very interesting that both of these diseases cause erosion of the bone. And this is what it looks like in the mouth. So we should have, between our teeth, this bone that holds the tooth together. It should not be a nice, tall, going almost to the, halfway up the tooth, and a rounded top. When we see early periodontitis, this bone starts eroding down. Finally, when you lose this much bone, the tooth starts to get loose. And people, usually older people who have advanced periodontitis, lose their teeth. So it's again like arthritis, it's a pretty debilitating disease. This is an x-ray that shows you the same thing. So see how you should have this nice bone coming up here? It's all cut off here. It's been eroded away, this person with periodontitis. And what about arthritis? The same thing is happening. This inflammatory response, starting in the soft tissue next to the bone, in the fluid that's next to the bone, then goes into the bone and causes erosions on the sides of the bone. This is what we look at all the time in the rheumatology clinic. This is a person who has arthritis in this joint. It's called the proximal interphalangeal joint. And the erosion of the bone on the sides, where the soft tissue connects to the bone. This is a close-up. So it's actually holes in the bone caused by inflammatory cells. This is another example of the side of the bone that's being eroded away. And you can see they've lost all the soft tissue in between the bones here, so there's no joint anymore. This is what's going on in both the mouth and in the joint. You have what we think in the mouth is activation by these bacteria that are in the plaque. This is why your dental hygienist is always picking the plaque off your teeth, to get all these bacteria lower. So the bacteria produce chemicals that activate immune cells. They produce fatty acids, FMLP, and lipopolysaccharides that immediately and very powerfully activate macrophages that may be hanging around the area. Then the macrophages produce cytokines, and IL-1, interleukin-1, and TNF-alpha are very powerful immune-activating pro-inflammatory cytokines. So they activate T lymphocytes and also B lymphocytes. The B cells are the cells that make antibodies. They become activated in the area and make antibodies, many of which are specific for the bacteria that are here in the plaque and here in the fluid between the tooth and the jaw. Some of these cytokines, like IL-8, attract other cells. This is supposed to be a blood cell coming into the area, attract other cells out of the blood into the tissue. And that's what we call inflammation. When you have this cell swarming into the area, they bring the fluid, they cause the redness, the swelling, the pain. Exact same cells, exact same cytokines in the joints and in the mouth. So it's not only the same cells, they're monocytes, neutrophils, and B cells, but the same genes. So this histocompatibility leukocyte antigen, HLA, DR4 gene, these are the genes that we say he needs a kidney waiting for a match. These are the genes that we say match for kidney transplant or bone marrow transplant. They're also extremely important in autoimmune diseases like arthritis. So DR4 is a gene that if you have arthritis and you have DR4, then you're going to have very severe, very bone erosive arthritis. You also have a higher chance of having periodontitis just by having this DR4 gene. The Pima Indians have a very high rate of rheumatoid arthritis. This is back to where this disease comes from. Our Native Americans here have very severe, very erosive arthritis. And that's maybe related to the DR4, but related to some other Native American genes. They have arthritis and they also have periodontitis. They're the same cytokines. This TNF-alpha and IL-1, these are the cytokines that we actually target in treatment. So some of these biologic treatments, you guys have heard of Enbrel or Remicade, they're actually antibodies or proteins that block TNF-alpha. And so it's a major cytokine that activates inflammation in arthritis. It's a super good drug. We have another one called anakinra that blocks IL-1, used for arthritis. Normally, these cells and cytokines, they have a purpose, right? Your immune system is supposed to be able to activate in case you have an infection. So people with severe infections have a lot of TNF and IL-1 too. But it makes you think about infection perhaps triggering the same immune mechanisms that we see in joints and mouth. Rheumatoid arthritis patients have worse periodontal disease, and patients with periodontitis have a fourfold increase of getting rheumatoid arthritis. And thinking about other triggers, not just infectious triggers, but smoking. Smoking increases your risk of rheumatoid arthritis and periodontitis. So what is going on? Is this all just a coincidence between periodontitis and rheumatoid arthritis? That's what we want to know. So we go back, you guys have learned about Koch's postulates, and this isn't just something you have to learn in class, but we actually use these postulates in real science. And as you remember Koch's postulates, in order to be considered for the cause of disease, a microorganism must be found in abundance in organisms with the disease, but not in healthy controls. So I'm thinking about maybe an organism that causes periodontitis and arthritis. What kind of organism would be found more in these patients? It has to be isolated, cultured from an organism with the disease, and then it has to cause the disease when inoculated into healthy controls. Remember that the H. pylori and peptic ulcer story where that scientist in Australia ate the H. pylori? He was going for the third Koch's postulate. And then you have to be able to re-isolate it in identical shape and form from the organism. So people have approached this looking at one of the oral organisms that is associated with periodontitis called Porphyromonas gingivalis. And the experiment is a mouse experiment because we can't really do this in humans very well. But they took a mouse that had a genetic background that made it already prone to arthritis, so similar to the DR4 background. And in fact, there is a mouse where they put the human HLA DR4 in it, and we've done this type of experiment. And then they can give the mouse oral bacteria. So they dip a Q-tip into P. gingivalis and rub it on the mouse's gums and teeth every day. And then they check for arthritis. So I don't know if you've ever seen arthritis in a mouse before, but this is what the mouse paw should look like. And they get extremely swollen joints, very painful, very red, so red it is. And if you did an x-ray, you would see bone erosions in that mouse's bone. So there are things that you can actually measure to show that given oral bacteria, that mice had worse arthritis. And then we want to see if we can treat it. So people have done, this is human studies actually, taking patients with refractory rheumatoid arthritis and treat their mouth. So do a lot of oral hygiene, pick the plaque off, give them an antibiotic with swishes, Sonicare, and so forth, and their arthritis improved. So that's just a relationship between what's going on in the mouth and the nose. But there's also evidence the other direction. So there's this funny Air Max mouse, which which is a kind of a mouse that has a deficient immune system. And in this mouse, a guy named Trombone induced arthritis with pristane. So this is a lipid that activates immune cells and squirts it into a peritoneal cavity. And that causes inflammation, it activates macrophages here, and then they go to the joints and they cause arthritis. And when it caused arthritis, then the mice got periodontitis. So it works both directions. You can induce arthritis with periodontitis, you can induce periodontitis with arthritis. So where does it start? What's interesting is they found that the mice required normal oral flora. So if they had them do antibiotic swishes, then they didn't get arthritis. And if they just gave them pathogenic bacteria like P. gingivalis, it didn't induce arthritis. So you needed a pathogen, and you needed normal oral flora to constantly trigger the inflammation in the mouth in order to get the arthritis. So, it seems the Koch's postulates may be partly fulfilled in the mouse experiments. What about the children? What's going on in the children? So we go back to the scientific method. Ask the question, I want to know what causes juvenile arthritis. Do background research. We've studied the adult rheumatoid arthritis literature, see what's there, and then construct a hypothesis. Then we test the hypothesis, analyze our results, and then go back to the drawing board and make a new hypothesis. So my hypothesis was that there may be a bacteria in the mouth of children that causes arthritis, in which case we have a chance to treat the arthritis by treating the mouth. The problem is I couldn't test this hypothesis because I don't know anything about the mouth. In medical school, they don't teach you about teeth and gums and the whole biology of the mouth. We don't do feet and we don't do mouths because we have dentists and we have podiatrists who take that part of medicine. So I knew very little about how to measure gingivitis and how to measure periodontitis and what the biology of the mouth was. But luckily, I happened to be at a luncheon where an old friend of mine, Peggy Lee, here, who had been in the lab with me 12 years prior, showed up. It was a reunion luncheon from our lab days. And she was a postdoc in immunology in those days. But when I met her a couple years ago, she was at the dental school at UW and working fully as a dentist and doing a little bit of dental research. So I started asking Peggy about this question, and she was sort of interested, but she was really happy to be a dentist. And she consulted with me a few times. I took her out for sushi and beer a few times. It took me about a year and a half. And finally, she's agreed to rejoin and get back into research. So Peggy Lee has been my dental component of this research project, and she's been invaluable in connecting us with dentists at the dental school and helping us to understand about what may be going on in the mouths of children with arthritis. So what Peggy taught me was that 100% of kids have gingivitis by the time of puberty. That surprised me. And we don't really know why. Is it biology? Is it hygiene? Is it going to bed without brushing your teeth? What is it? But we see a surge of arthritis around this same time in children, so it makes you wonder if it's connected. Juvenile arthritis patients with gingivitis seem to be difficult to control. Kids with, there's one paper from Iceland suggesting that kids with juvenile arthritis do have worse gingivitis. Peggy told me the methods were flawed and they weren't testing for the gingivitis and measurements of bleeding on probing well enough or something. And she was not pleased with that, not convinced. And so our question is, does gingivitis trigger juvenile arthritis? And if so, then we should see similar immune responses in those two kids. So we should be able to look at their cells, look at their cytokines, and they should look similar in kids that are healthy with gingivitis versus kids with juvenile arthritis. So one of the things we can look at, we can look at cell markers. We can also look at autoantibodies. And the autoantibodies we're targeting are these antibodies to citrullinated peptides. And the reason why we're focusing on these is because these antibodies are only found in people with arthritis. There are a lot of autoantibodies, like anti-nuclear antibodies, or antibodies to our own nuclear components, that a lot of healthy people have too. These anti-citrullinated antibodies are only in people with arthritis. And what they are antibodies that recognize a modified peptide. So the peptide has to be made first, and then this enzyme called peptidyl arginine deaminase, or PAD, deaminates the arginines and turns them into citrulline. So you have a charged amino acid that turns into an uncharged amino acid. And the result to the immune system is that this was formerly thought of as part of a peptide that belongs to self. After this process, it no longer looks like self and can activate the immune system. So we can make antibodies specifically recognizing only these citrulinated peptides. So this happens during inflammation. Inflammatory process can induce one of these PAD enzymes in our own cells, and we can citrulinate our own proteins. So anytime you get an infection, you get redness, swelling in the area, there's probably citrulinated proteins being made and then broken down. And we see it, we think these antibodies may actually cause arthritis. And I'm going to connect it to the bacteria back in a second. And the reason why we think that is these are patients with rheumatoid arthritis, and these are these antibodies that I told you only happen in patients with rheumatoid arthritis, but this is 10 years before they were diagnosed. So they can get these blood samples from military recruits who give blood as soon as they join the military and every year afterwards. So if they get arthritis 10 years later, you can go back and get their blood out of the freezer and test it every year. So that's what they did, and they found that a lot of these patients were making antibodies first, and then 5 years later, 10 years later, developed arthritis. So it doesn't prove it, but it suggests that these antibodies may cause the arthritis. And somebody else, of course, did a mouse model where they injected these antibodies into the mouse, and the mouse got arthritis. So they're bad antibodies. This is Becky Hausman. She did her PhD in immunology at Berkeley and was working in the lab, and she started looking at this question, trying to figure out what about oral bacteria connects these anti-citrulinated antibodies. And what she came up with is that only Porphyromonas gingivalis makes the PAD enzyme, the only bacteria that makes this enzyme that's able to citrulinate proteins. So it's a pathogenic bacteria. It's a major cause of periodontitis. It can increase antibodies to P. gingivalis in rheumatoid arthritis patients, adult rheumatoid arthritis patients, correlates with increased antibodies to CCP. And we have a CCP antibody test in clinic now. When a patient comes in and has severe arthritis, I'll just order that anti-CCP antibodies in the clinic. So these antibodies can cross-react. So could it be that this P. gingivalis was activating the B cells to make anti-citrulinated antibodies, and then they travel to the joints and they react to the citrulinated antibodies in the joints? So this is what Becky suggested may be going on, that there's a child who has a genetic susceptibility. We don't know what that is, but maybe something like the DR4. And they get infected with P. gingivalis. So we're not born with P. gingivalis in our mouths, but they get spread through families or other kinds of mouth-to-mouth contact. And most of us have been exposed by the time we're 15 or 20. Then P. gingivalis, the PAD enzyme, citrulates human proteins in the mouth and induces antibodies to the P. gingivalis citrulinated proteins and to the human citrulinated proteins, to our own citrulinated proteins. And these antibodies bind to bacterial proteins and human proteins, both from complexes, and these complexes go into the joints to activate. We know that the complexes of antibodies and antigens, they form these big large molecules that can activate macrophages. So how do we test that? Claire Murphy is a student at Georgetown. She was a sophomore when she came to the lab last summer, and Claire took this on. She wanted to measure the anti-CCP antibodies in the children with juvenile arthritis and compare that to the CCP antibodies in controls. And then she wanted to see if the CCP antibodies in these kids were cross-reacting to P. gingivalis proteins. So we can have peptides made that look just like P. gingivalis proteins. We can have them citrulinated or not citrulinated, and we can take human homologs of that same antibody and say it's citrulinated or not citrulinated. So it shows up a protein that we know is a target in some adults with these diseases called enolase, and we know it's citrulinated. And P. gingivalis has an enolase, and we have an enolase. Here we have a human enolase. And the PAD enzyme can citrulate either enolase. So how we tested it was with an assay called ELISA, enzyme-linked immunosorbent assay. This is to test the antibodies that are specific for our enolase citrulinated proteins. How the assay works is we put the P. gingivalis antigen, or the human enolase antigen, on the bottom of the well. So there's this 96-well plate, and this is one of the wells in 96-well plate. And then we put the patient's serum in, and if there are antibodies in the serum to this protein, they'll stick. And then we put another antibody, an anti-IgG, that will bind to any human IgG molecule. And this has an enzyme attached to it. We put in a substrate which turns color when this enzyme is present, and only wells in which there are antibodies to the antigen will light up and become colored. So this is what it looks like, the wells look like from the side. So we have, this is a positive control and a negative control, so a person that definitely has these antibodies and someone who definitely doesn't have these antibodies. Here's our antigen, the enolase protein on the bottom of the well. This is the patient's serum. So this person had antibodies, so they're sticking. This one doesn't have any antibodies. They have antibodies, but they're not sticking to this protein. They're not specific to this protein. Wash it all away. The secondary antibody, there's a little enzyme, and we put it in the substrate, and it turns blue. So this level of blueness, there's a machine that measures how intense the blue is. It's really intense, and this patient had a lot of these anti-enolase antibodies. If it's not blue at all, then we haven't had any antibodies at all. So that's what the plates look like. So Claire ran these assays on 21 patients with juvenile arthritis and 21 controls. And you can see the controls didn't have much at all in the way of these antibodies, and that's what we expected. So it's a really good assay. The negative controls worked. And normal children, who haven't really been tested for these antibodies, healthy children, this has been done in adults a lot, but not in children. And children make a lot of antibodies because they're always getting infected, right? They're always getting ear infections and tonsillitis and stuff. So their immune systems are always souped up. But they're not making antibodies to citrullinated proteins at all. And patients with juvenile arthritis that have antibodies to citrullinated proteins at all, so this is our clinical test, it's a whole bunch of peptides that are citrullinated, are these triangles. Patients that had those also made antibodies to human enolase and to the P-gin-divalis enolase in the dark squares. The light ones are the uncitrullinated antibodies, so they never made them to the antigens, peptides. So they're only making antibodies to citrullinated peptides. So we can look at a correlation now. So Claire did correlation tests. Patients that had anti-human CE citrullinated enolase peptides also had anti-P-gin-divalis citrullinated enolase peptides. So this suggests that P-gin-divalis in the mouse is inducing the production of these antibodies that then bind to the cell proteins that can then form complexes and go to the joints and arthritis. She looked at the clinical test that we do. This is a bunch of citrullinated peptides, and they correlated with the P-gin-divalis. So what we're testing in the clinic could very well be an immune response to P-gin-divalis in the mouths of these kids. So here's the question: can kids with gingivitis mount systemic inflammatory responses? How is this happening? Why doesn't everybody get arthritis? If a lot of us have inflammation or gingivitis, 100% of teenagers have gingivitis. Why are they not all getting arthritis? So there must be something about the immune response. And we're interested in the very primary immune response to bacterial infection. Could P-gin-divalis infection in kids with certain genetic background progress to make CCP antibodies and then develop JIA? So we made two hypotheses. One, that gingivitis correlates with active juvenile arthritis. And two, that JIA and gingivitis provoke the same inflammatory response. So activate the same cells and induce the same CCP antibodies. And the first thing you do when you make hypotheses and you want to test a new hypothesis, you have to write a grant. So Peggy and I wrote a grant, and our Arthritis Foundation gave us $200,000 to test this hypothesis in children. So I told you about the cytokines and the macrophage activation early on, but I didn't really talk to you about the cell markers that we're looking at and how do you tell if a cell is activated. So this is picture of T cells actually attacking a tumor cell. These white ones are T lymphocytes attacking a tumor cell. We know that cytolytic T lymphocytes are made to kill cells, kill cancer cells, kill host cells, attacking virus. So this happens normally in our immune systems all the time. In autoimmunity, T lymphocytes attack our own cells. So people with arthritis have T lymphocytes that would be attacking their own cells in their joints. So obviously causes tissue damage and is not good. So the T cells also produce cytokines, which activate macrophages and neutrophils. So it's all together in this whole big inflammatory response. So when we get an infection, this has to happen. We have this big inflammatory response, but then we have to turn it off. Otherwise, the inflammation will just go on forever. So every time you get an infection, the immune system turns on and turns off. So how does it turn off? The white blood cells or cancer cells that activate the T cell produce proliferation and cytokine production, and then they have to turn it off. So one of the molecules that are produced by both cancer cells and activating cells is called programmed death ligand 1. Programmed death because it actually kills T cells off. So T cells will recognize this programmed death ligand 1, and it turns them off, it stops them from making cytokines, it stops them from proliferating. It's an inhibitory molecule. We found that some patients with autoimmune diseases or chronic inflammatory diseases can't produce much PD-L1. So they're not able to turn their immune system off. So how do we test PD-L1? We get a blood sample, and we use these special tubes called cell filtering tubes. And this is a gel layer here, so we put whole blood here. And we spin the centrifuge for 20 minutes, and all the red blood cells and the neutrophils go down to the bottom, below the gel, and the white blood cells, including lymphocytes, NK cells, and monocytes, are all above the gel. And this is the plasma on top. So we can take the plasma off, look at antibodies, study antibodies in the plasma. We can take these white blood cells off, and we just suck them off with a pipette. And we can study the white blood cells by flow cytometry. In flow cytometry, we have specific antibodies for whatever protein we want to look at on the surface of the cell. So in this case, we want to look at PD-L1 expression on the cell. I have a specific anti-PD-L1 antibody with a fluorescent marker, fluorescein green. And then we suck them into the flow cytometer, where a laser shoots at each single cell individually. And if the cell has a fluorescent marker on it, it will emit a fluorescent signal, which can be recognized by the detector. So where my lab is in Seattle Children's Research Institute, we have machines that have 5 different lasers and 16 different detectors. So you can imagine we can on one cell identify up to 200 different types of cells by the combinations of different colors. So you can have purple fluorescence, blue, and green, red, orange, and so on. So using this method, we can add a marker for monocytes, a marker for activated monocytes, a marker for PD-L1, a marker for T cells, and this one blood sample look at expression on all these different cell types. This is just the PD-L1 on the monocytes. And what we found is that, this is for a study that I'd done before this whole device thing came up and made me interested in this molecule PD-L1, is that normal people express a little bit of PD-L1. So this is the number of cells plotted in relationship to the intensity of fluorescence on each single cell. So it tells you the level of PD-L1. So people normally have about this much PD-L1. Kids with infection have a huge amount of PD-L1 on their surface, really activated cells. Patients with lupus, an inflammatory autoimmune disease, don't express much PD-L1. So a whole other project in the lab is why don't they express PD-L1 and how does this let their inflammation get out of control? When we treat them, with steroids, chemotherapy, and anti-inflammatory medicines, they can express PD-L1 again. So it's a highly regulated gene related to how much inflammation is in your body. But what was interesting about these people with lots of infection and lots of PD-L1 is that we tested 20 kids with JIA, and they had huge amounts of PD-L1 on their monocytes. So the kids with JIA looked like kids with infection, but they didn't have infection that we knew of. And this just shows you the population. So these are control, healthy people. Patients with lupus, active lupus, a little lower, it's about 5-fold lower. This is a log scale. And the patients with JIA, on the log scale, they're 10 times higher in their PD-L1 expression. So they look like kids with infection. So the specific hypothesis we're testing is that gingivitis correlates with active arthritis in JIA, and that PD-L1 is high in cells from patients with gingivitis and no JIA. So here's what we designed a trial. We got together with a statistician and asked him how many kids do we need to test to prove this. and decided that 50 kids would be good. How are we going to find, for controlled, and 100 kids with AIA, how are we going to find 50 children who will give us a blood sample? That's our next obstacle. So we finally decided that when kids are asleep, they don't mind if you take blood from them. We have a dental clinic in Madison Park, it's a pediatric dental clinic, where kids go to get procedures under anesthesia. They're going to get their wisdom teeth out, or some of them just have a whole bunch of cavities, and we have to put them to sleep in order to fill all their cavities. So while they're asleep, we get a blood sample from them. They're not 100% healthy, they have problems in their mouths, but they're not inflammatory problems generally. Or so we thought. And then 100 kids in the juvenile arthritis clinic and the rheumatology clinic, they give us blood all the time because we have to test their blood to monitor for their medication side effects. So they're kind of used to it. So we just get a little extra tube of blood when they're getting their regular blood for their clinic visit. Every kid gets a dental exam. So we have Peggy Lee or one of her friends do a very thorough dental exam. And then the kids in the arthritis clinic have a joint exam to see how much arthritis do they have on that day taking their blood. So then we can correlate how much gingivitis, how much periodontitis, how much arthritis, and how much PD-L1 in blood, and how many CCP antibodies can we detect. So how do we get these samples? This is Gretchen Hensdorf, and she is our clinical research assistant. So Gretchen is in charge of doing all the human subjects protocols. So we have to get this approved by the Human Subjects Committee at Children's Hospital to make sure that we're not hurting the patients. And then she's in charge of enrolling the patients. So she goes to the clinic and she invites the families to participate. She informs them what the study is and gets their consent. And then takes the patients to the lab, gets the blood, takes them down to the dental clinic, gets the dental exam, collects all the paper, makes sure that everybody filled out their papers correctly. And this is an example of some of the papers that are filled out for this study. So has anyone been in a clinical study before? Couple people have been. So they usually give you a stack of papers to fill out, questionnaire and check boxes and so forth. So we have a lot of those for this study. Because there are a lot of things that might affect inflammation in the mouth, like do you have braces? Sorry, pushing the wrong button here. There we go. Problem with your jaw. If you have jaw problems, you might not you self-cleaning your mouth as much, if you're not moving your mouth very much. You have any tooth pain, this is inflammation in the eyes associated with arthritis. Are you a mouth breather? People who are mouth breathers may not have as much saliva to clear out their mouth and keep the bacteria washed out. And then how do you measure arthritis? We need to have numbers to correlate. We can't just say there's a lot of arthritis or a little arthritis. We actually have to have precise measurements in order to make correlations. So do we take the number of joints? Do we take a pain score? Do we take disability? How much disability? How do we do that? So I always turn to the experts when I have these questions. And yeah, I'm a rheumatologist, but I don't do arthritis outcomes research is what we call this, how to measure how much arthritis. But I have two colleagues, Dr. Ringgold and Dr. Wallace, who are world expert in this area. In fact, they've been involved in this whole project of defining what is active arthritis in children. And how do you measure the level of arthritis in children? So I get advice from them, and we developed our protocol. There we go. So this is what the doctor fills out now. We have our homunculus here, and they fill in any joints that might have swelling, or redness, or pain on motion, or restriction of motion. And then they fill out whether the patient has antibodies called rheumatoid factor or CCP antibodies, and how are they doing in general? Are they flaring? Are they a little improved? Do they have morning stiffness? That kind of thing. The doctor that sees the patient in the clinic has to fill that out. And then, how do you measure gingivitis? I knew nothing about that. The dentist kicked in, and we have 4 scores for the mouth exams that the dentist has to fill out. One has to do with how much tooth decay they have, how many teeth are missing, how many fillings. And imagine if we enrolled 6-year-olds, 7-year-olds, there'd be a lot of missing teeth, but it would have nothing to do with periodontitis. So then we had to change our enrollment plan to only kids that already have all their teeth. So we only take 12-year-olds to 18-year-olds. So we're missing that whole younger group of kids because there are no good dental outcomes to measure. The plaque index, how much plaque is on your teeth, how clean are they along the margins, and the gingivitis index. So here's an example of early gingivitis. See the erythema around the teeth, and they're kind of swollen around there. If you probe this with one of those metal probes that the dentists use, then there would be bleeding around it, the area. So they have this whole gingival index, which they score totally normal, mild inflammation with slight color change, no edema, no bleeding, and they're really big on this bleeding on probing thing. And that's what our hygienists are always doing, probing, and that's why they're doing it, because if there's bleeding, that means there's inflammation in that area. Who knew? So this is why that's important for not only gingivitis but also periodontitis, and this leads to the periodontal index. So this is healthy teeth and gums. They probe in between the teeth and the gums. So if they just can probe a little bit and there's no bleeding, then that might be a little bit early gingivitis. You shouldn't be able to probe at all. So they have a measuring, this is like a measuring stick on their probe with millimeters. And early, moderate, advanced periodontitis, they can stick the probe deeper and deeper and actually measure that. So we have a number. This is called the Community Periodontal Index, which WHO uses all around the world to measure how much periodontitis people have. And they have to do it in 6 different teeth. So this is a lot of data we're collecting from all these sheets that the doctors, the dentists, and the patients fill out. What do we do with all this data? Well, we have Leo A.G., who's actually a new student, they're a sophomore. He's our database manager. And we enter it all into this awesome online system called REDCap, which is developed in Vanderbilt, and we use it in several different universities around the country. And we can design our own study and put whatever fields we want in. So she's made this community periodontal index field, and she can just enter the numbers in there for every patient, and then can easily export Excel files with all the data. It just spits it right out. So that's a whole job that Leo has in organizing our data. This is the preliminary results. You guys are the first to see this because we're only halfway through this study. We have 102 patients total recruited, so we have 48 more to go now. And our preliminary data looking at the gingival index is quite surprising. What it looks like is that the controls have worse gingivitis than the JIA patients. So we go back to the, test the hypothesis, look at the results, analyze the results, and then think again. So we've been thinking a lot, and we organized several meetings with the dentists and the rheumatologists, and trying to figure out why this might be. And one thing that came up was that before the kids come in for anesthesia, they're told not to brush their teeth for 24 hours. Who knew? But it's okay, because we can still correlate the PD-L1 expression with the extent of gingivitis. We can still test this hypothesis that if you have inflammation in your mouth because you're not brushing your teeth, you may activate the immune response, which can be detected in your blood. So, but it does not look like the arthritis patients have worse mouths. Another hypothesis that we can test is that these juvenile arthritis patients are treated with immunosuppressive medications. So they're taking naproxen, ibuprofen, stronger immunosuppressive like the TNF inhibitors. So if we're inhibiting the TNF in their joints, maybe we're inhibiting the TNF in their mouths as well. So you don't see the inflammation because we're blocking it. We have all the data in order to test that. There's a plaque index. You see it's a little higher in controls. And if you do beams and students t-test, compare the two groups, then you see the controls are actually higher in the periodontal index as well. So how can we test whether the immunosuppressive medications actually influence this? And are there other factors in the lives of the controls that may influence their oral health? So we collect other data like cigarette smoking. I told you cigarette smoking increases the risk for periodontitis and arthritis. So we have a confidential sheet that is an envelope handed to the child fills it out, puts it in the envelope, gives it back to us. So the parents never see it. And they should be free to report their smoking habits on this page. The parents also answer questions about smoking, because secondhand smoking might affect inflammation and activate oral inflammation. And then we look at socioeconomic status, how much school the parents have, what is their household income, which some people are not comfortable with answering, it's fine, but from the data we have, it's possible that the patients that come from houses of lower socioeconomic status have less dental care and may have more oral active inflammation. So we're able to test that, but it gets to be sort of a complicated statistical problem at this point. You have to do a multivariate analysis and to ask these types of questions. And I don't know how to do that, so we call in another specialist, and this Brian LaRue, who is a statistician in the dental school. So he's used to asking these kinds of questions, and he knows how to do these multivariate analyses to look at all the factors that may be influencing our periodontal scores and also our PDL1 expression assays that we do in the lab. So back to Koch's postulates and the problems that we now need to think about in using Koch's postulates, which are partly, for some of these reasons that are stated here, may be becoming a bit obsolete. So the first one, that the organism be found in abundance in organisms with the disease, but not in healthy controls. The problem is that many pathogenic bacteria are found in healthy individuals. So we have a Vibrio organisms in our mouth, but we're not safe with it. And the host immune response determines pathogenicity. Lots of people exposed to tuberculosis organisms but don't get TB. People exposed to HIV but don't actually get HIV. So it depends on the host immune system who gets sick and who doesn't. The organism needs to be isolated and cultured from a person or an organism with the disease. But we know now through gene sequencing, this whole microbiome field where you sequence everything in the mouth, 80% of what is in your mouth does not belong to you. It belongs to some sort of bacteria, and only 20% of those can be recognized as known bacteria that grow on our agar dishes, that grow on our blood agar. Whenever we can culture conditions that we create, the bacteria in our body don't care about our dishes. So there's lots of bacteria that we haven't even discovered yet. You're not necessarily going to be able to grow it in a dish. Should the bacteria has to cause the disease when inoculated at healthy controls, again, this host immune response determines whether you get sick or not necessarily the organism. And it needs to be re-isolated in the same form and shape and grown. 80% of our bacteria in our bodies have never been cultured. So the questions I'm going forward with are that the oral microbiome in children with juvenile arthritis may be different than in gingivitis and than children with no arthritis. So we can look at this by getting samples from the plaque in the children's teeth and sequencing everything in it and comparing that between the children with arthritis and the children with gingivitis and the children without gingivitis. There's been a pretty big, it's actually the National Institutes of Health has a whole program on the microbiome now. Because we think that the 4 pounds of bacteria that we all carry in our bodies are probably influencing our immune systems all the time and maybe related to a lot of the diseases that we think of are autoimmune diseases. You've probably heard of some people with arthritis are connecting it to inflammation in the gut. So what they eat, called celiac disease, where people are allergic to meat, they can get arthritis as part of that. So inflammation in the gut is related to inflammation in the rest of our bodies. Then there must be a genetic basis for this immune response to oral bacteria. So we can do sequencing of the whole genome of these kids in this study. We have DNA from all of them now. So we can look for specific immune-related genes. We can do the whole genome sequencing, or we can do what's called an immunochip. So somebody's made a chip of polymorphisms, 50,000 of them related to the immune system. So for like $100, we can run this little chip and see if there are any changes in the immune system-related genes in the kids with JIA versus the kids who don't have JIA. And then we're going to go on to look at the systemic immune response to the oral bacteria. And then we can think about interventions. So if this really is an infection, so gingivitis, and it reduces the PDL1 expression on cells in the kids with those underlying genes, then can we treat this arthritis with topical antibiotics? Can we do a chlorhexamide wash in our mouths and get rid of that bacteria, and then the arthritis will get better? Or do we need to replace this pathogenic bacteria with good bacteria? Kind of like the lactobacillus thing, people eat like yogurt, it's supposed to give you good bacteria in your gut. Maybe that needs to be done in the mouth, so you have competition for the pathogenic bacteria. And then does this pathogenic bacteria problem extend into the mouth? And the mouth is really the beginning of the GI tract, right? It's all connected, so does it extend down into the GI tract? And what would be really cool if we could do this intervention study. Well, we give the kids a Sonicare, or we you can go to the dentist every 3 months for a really good cleaning. And that blocks the P. gingivalis or the other bacteria that is discovered, is more important in children, that are citrullinating proteins and then inducing this antibody-inflammatory response. So that's the story for arthritis. Arthritis is not the only disease that has been associated with gingivitis and periodontitis. This is a whole list of diseases that people think are linked to inflammation in the gums and in the bones of the mouth. And the one that has the most data is cardiovascular disease. So there's a very strong link now between atherosclerosis and periodontitis. And it's still, we think it has to do with the oral bacteria triggering the immune response, but also there's a big genetic component. And interestingly, rheumatoid arthritis patients tend to die of cardiovascular disease at an early age. They have very advanced cardiovascular disease. So if we could approach this by just treating the mouth first, we could avoid all these immunosuppression medicines and all the cardiac outcomes. So this is our group in the lab. I told you about Becky Hausman, the immunologist, who's now working on the science bus that Children's Hospital runs. It's a great big laboratory bus. She loves to teach. And they go around to middle schools and elementary schools around the state of Washington, and kids get on the bus and they do experiments. They isolate DNA and they get cells, and they have science day on the bus. Jess Foster is our lab manager, and she runs the lab setting along with Claire, who's coming back for another summer this year. Ed Kim, another undergraduate student, and Leo, the database manager. And these are our clinical collaborators, the dentists and the rheumatologists. So you can see it takes a lot of people. I left the statisticians off of here. I should add them on because they're an essential part of this team. And then our funders, of course. And this is a part of our group. So I'd love to hear what questions you guys have about this area or rheumatology.
Any questions? Dr. Stevens.
Yes.
So how long now have you been working on just this project with JIA and T?
This is a very new project. We've only been working on this, well, the grant started a year and a half ago. So only a year and a half. This is a very fast, rapid project.
Yeah.
My other project, working on maternal cells in children with lupus, has been going for 12 years now. And we're just publishing a paper this year. So it varies. Sometimes it takes a really long time. These types of assays are not too hard. And we're ready to do the assays because of the work we've done over the last 10 years. We're ready to go.
Yeah, it seemed like you drew some conclusions from both the lupus, talking about the PD-L1 lupus and JIA. Did you find any other conclusions between the two? Are you seeing any more similarities?
Comparisons between the arthritis and the lupus patients are really interesting because almost every marker we say, SNE, in lupus is opposite of every other autoimmune disease. And we don't understand this at all, but there are other autoimmune diseases like multiple sclerosis, diabetes, as well as adult rheumatoid arthritis, where, for instance, the PD-L1 is very high. In lupus, it's low. Different kinds of cells. It's the cells that produce IL-4 in lupus, the cells that produce interferon gamma and TNF in arthritis. The patients that are treated with these TNF blockers for arthritis can get lupus as a side effect. So it's fascinating. They're both inflammatory diseases. Both cause arthritis, but the arthritis in lupus doesn't cause bone reviews. It doesn't affect the bones, it's just the tissues in the joints. Fascinating. I mean, it's completely different directions of the immune system. Yes?
Early on in your talk, you talked about the correlation potentially between sugar and its introduction and rheumatoid arthritis. I was wondering if you, on any of your many questionnaires, had questioned the subjects about their diets and seen any sort of correlations there.
Yeah, we've thought about that and really struggled with how to ask that and how to collect that data. on the diet. And so the question we have now is, do you drink sugary drinks? Which is really hard to measure. You know, should we say how many Cokes do you drink per week, or what would be the best way to turn that into a number? And then we also collect the weight and height, so we can do a BMI on the patients, which is a giant leap from BMI to bad nutrition, but it's maybe a marker of it. I haven't pulled the nutritionist in to the team yet. Thank you for giving me that idea. Probably what we should do. For the smoking questions, we consulted the adolescent medicine experts who do a lot of research on smoking, so they know how to ask those questions and how to word them exactly right so that you get information that will be useful. So we just said, do you smoke or not? What does that mean? Yeah, I smoked once 10 years ago, or I smoked a pack yesterday. So you have to be very precise in asking those questions. And it probably makes a difference. We asked about sugary drinks, and we asked, did you brush your teeth in the last 24 hours? So that we can have those comparisons and actually do subgroup analyses to see if it makes a difference.
Was there a particular experience in your undergraduate years that led you to pursue the pathway of an MD PhD program? So was there anything that what led you to want to pursue that?
I started out as a chemistry major, and the most interesting part about chemistry was the biologic part of it. I like to learn how things work. It was fun making molecules, and I worked actually at a pharmaceutical company, Syntax, for a summer, making a heart drug. So it was kind of fun. But I really liked understanding how cells work and how bodies work and how things fit together at the molecular level. So I looked around UC Davis to find a lab that I could work in, just to get some experience. And one lab was taking undergrad students and happened to be a rheumatology lab. So they started talking, the PI and the principal investigator of the lab would sit the undergrads, the three of us, down and give us these lectures about what we know about rheumatoid arthritis. And there was this huge black box in the middle, and it's still there, the black box, but we filled in the sides of it. So it got to be interesting. And then there's a human side of it, in that shortly after that, my grandmother, who had been a super healthy lady, gardening, really active, got horrible rheumatoid arthritis. And all of a sudden, her knees were this big, and she couldn't get out of the car. And it happened a year after Grandpa died. And so I started thinking about stress relationships between the immune system and the brain, and started learning a lot about how our brain, our nerves actually affect our lymphocytes. And then she developed lymphoma, and it turns out lymphomas are more common in patients with rheumatoid arthritis. So it's all connected. And I felt that was a good problem to work on. That I could see the change in quality of life of a person from a disease and how it can change so quickly. And that's the wonderful part about rheumatology right now, is we have these great medicines like Enbrel, so we can bring people with severe arthritis like that right back to normal. We get kids with swollen knees like that, when I showed you, back on the soccer field within a few weeks with the right treatment. So it's a very satisfying field to be in for humanitarian reasons, and also science. As you can see, there's a lot of questions to read.